Mitosis is the process by which nuclei divide. Mitosis is essential for the growth of organisms, splitting one cell with a full set of chromosomes, strands of DNA, into two genetically identical daughter cells. This allows organisms as a whole to grow larger, allowing for the growth of numerous cells with identical genetic compositions, allowing each and every cell to grow and function as an individual.
Meiosis is the process by which organisms create sex cells, which are essential for any form of sexual reproduction. Even hermaphroditic organisms, which do not have any visible divisions of sexes, require meiosis to produce two types of gametes, which are sex cells, such as sperm and eggs, that join to form a zygote. Meiosis allows these gametes to be intercompatible, leaving daughter cells with only half the normal number of homologous chromosomes. Therefore, the zygote has the correct number. Without the capability to reproduce sexually, organisms have very little variability, barring evolved methods to mitigate this disadvantage. One very important part of meiosis is independent assortment. Independent assortment states that chromosomes are separated at random, independent of each other during meiosis, allowing for increased genetic variation.
In interphase, the chromosomes are being copied. Each chromosome undergoes DNA replication, creating two sets of chromosomes to be divided during the first half of meiosis, during which there will be two nuclear divisions resulting in 4 gametes. Prophase 1. During this stage, the chromosomes condense and become visible inside the nucleus. Since the chromosomes were replicated in an interphase, each now has sister chromatids joined at the centromere, creating an X shape. The chromatids in each chromosome pair align next to each other. After the alignment, a process called synapsis occurs, during which the two chromosomes in each pair become tightly connected along their lengths. Each pair of homologous chromosomes is called a tetrad. While synapsis is occurring, a trade of adjacent bits of DNA is occurring through a process called recombination. In the end of prophase 1, the nuclear membrane begins to break down. The spindle grows out from the centrosomes on each side of the outside of the cell. Prophase happens in the cell so that chromosomes are able to move to the center of the cell. Additionally, recombination can create higher genetic variability in daughter cells and prevent errors.
Metaphase 1. During this stage, microtubules sprout from the spindle and attach to the kinetochore that is close to the centromere of each chromosome. Microtubules from one side of the spindle attach to one of the chromosomes in each homologous pair, two chromosomes that code for the same traits, while microtubules from the other side of the spindle attach to the other member of each pair, the chromosome pairs that line up along the equator of the cell. This will allow them to be evenly split into new cells. Anaphase 1. The microtubules disassemble and contract, causing the homologous chromosomes to separate so that the two chromosomes in each pair are pulled towards opposite ends of the cell. This separation means that each of the daughter cells will have the number of chromosomes of the original parent cell.
Telophase 1. The new chromosomes reach the spindles at the two ends of the cell. The cytoplasm then reorganizes and divides, leaving two daughter cells. While the daughter cells have the same number of chromosomes as the original cell, recombination ensures that they are not identical. Interkinesis. There are now two daughter cells which reform their respective nuclear membranes. The chromosomes do not replicate during this period, as the second meiotic division will result in gametes with only half the number of chromosomes as normal cells.
Prophase 2. Because the spindle might have been disassembled in interkinesis, it reforms here. The chromosomes, like in prophase 1, condense into tight structures, and the nuclear membrane is once again disassembled. Metaphase 2. Metaphase 2, much like metaphase 1, consists primarily of chromosomes aligning in the center of the cell. Anaphase 2. Anaphase 2 occurs after metaphase 2. In anaphase 2, the chromosomes begin to be pulled apart and toward the opposite poles via spindle fibers.
Telophase 2. The new chromosomes, now separated, have new nuclear membranes form around them. The cytoplasm divides and 4 daughter cells remain with half the normal number of chromosomes. Recap. Now that meiosis is complete, let's go over what happened. We started with one normal cell. This cell replicates its DNA to prepare for the first meiotic division. After going through meiosis 1, we were left with two normal daughter cells. And meiosis 2, the two daughter cells go through the same stages as meiosis 1, with the exception of interphase, to produce 4 gametes with half the normal number of homologous chromosomes. Daughter cells in mitosis are completely identical to the mother cell. In meiosis, the 4 daughter cells each have a different genetic makeup and composition. In meiosis, the parent cell is a diploid containing a full set of chromosomes. The daughter cells, however, are haploid, containing half of a full set of chromosomes. Meiosis differs from mitosis mainly because it creates 4 daughter cells with different genetic composition, while mitosis creates 2 identical daughter cells. Fertilization takes place when an egg and a sperm meet. The egg and the sperm contain 23 chromosomes, each containing half of the genetic makeup of a normal human cell. When an egg and a sperm meet, the sperm enters the egg cell and deposits its chromosomes. Then, the fertilized cell, known as a zygote, contains all 46 chromosomes and begins division, creating a new organism.
Abadi, that's all, folks.